Good morning, dear students. So, welcome to yet another class of YouTube live classes in botany. So, today's topic for the class is respiration. So, we have completed the chapter on photosynthesis in the previous classes. So, photosynthesis was a unique process that was confined to the plants only, and the green plants are the organisms that are capable of carrying out photosynthesis. The next physiological phenomena that we observe in all the living organism is known as respiration. So, respiration is a phenomena in which the cell derives energy from the organic molecules that is included in our diet or that we take in as our food. So, today we shall discuss the basic characteristic features of what is respiration and what are the different types of respiration. So, let us go to the definition of respiration. So, respiration, as you can see, respiration is a common metabolic path that can be seen in all living organisms. So, respiration, how do we define respiration? Respiration can be defined as a process in which large organic compounds are broken into simpler molecules are broken down into simpler compounds with the release of energy inside the cell. So, here large organic compounds are broken into smaller inorganic compounds or simpler compounds. The energy that is released during the oxidation of the organic compound is trapped in the ATP molecule. So, here the large organic molecule include the molecules like our carbohydrates, also proteins as well as lipid molecule. The common organic molecules, however, are the carbohydrates or sugar from which we derive maximum energy. The energy is not ever released directly, but it is in the form of ATP molecule. The energy is trapped in the ATP molecule. So, this is a series of stepwise enzymatic reactions which leads to the synthesis of ATP molecule. So, what does the ATP molecule do? The ATP molecules serve as the energy currency. We call the ATP molecules as the energy currency. Why energy currency? Because in all the phenomena we calculate energy in terms of, say, suppose there is requirement of 2 ATP or 8 ATP. So, we take them in the numbers. So, it is called as the energy currency. The respiration phenomena, depending on the utilization of oxygen, it can be divided into the aerobic, anaerobic type of respiration or aerobic type of respiration. So, in the anaerobic type of respiration, there is no oxygen utilized. No O2 is utilized in anaerobic respiration. Whereas in aerobic respiration, there is utilization of oxygen for the oxidation of the food material. So, coming to anaerobic respiration. So, coming to the anaerobic respiration, we shall see what is anaerobic respiration and its characteristic features. So, anaerobic respiration here, as the name is indicating, it is without the use of oxygen. So, oxygen is not available. So, here oxygen is not available for the oxidation of the respiratory substrate like your carbohydrate. So, what is respiratory substrate? The substance that is used. So this substrate means substance that is used for the purpose of releasing energy. So the substance that is used is called as the respiratory substrate. The common respiratory substrate are the carbohydrate or sugars like glucose molecule. So it results in incomplete oxidation of the respiratory substrate because oxygen is not involved. So what happens in these cases is, suppose we take the 6-carbon sugar glucose. So glucose has 6 carbon. It is a hexose sugar or an aldose sugar with 6 carbon. So whenever oxidation is occurring, these carbon bonds shall break to release, and the released energy is trapped in the ATP molecule. So when oxygen is not present, there is a partial breakdown of the respiratory substrate. So if glucose is present, the partial breakdown may give you different types of products. For example, one is your alcoholic releasing anaerobic respiration or fermentation. The other is where lactic acid is released. So these two compounds, that is alcohol as well as lactic acid, are a result of partial breakdown or partial oxidation of the glucose molecule. For example, you see in case of the ethanol fermentation, which is carried out by the Saccharomyces or yeast cells. So yeast cells contain enzyme zymase, which help in the breakdown of glucose into ethanol and carbon dioxide. So in ethanol, this is C2H5OH. So we find that the carbon number is 2. So how many molecules are formed? There will be formation of 2 alcoholic groups or ethanol and release of 2 carbon dioxide molecules. So here this will become 1 CO2, the second CO2 and 2 alcohol will be formed. So this is the breakdown of glucose. So glucose gives 2 molecules of ethyl alcohol and 2 molecules of carbon dioxide. Similarly, when there is breaking of glucose into another compound called as lactic acid. So lactic acid is a 3 carbon compound. So you take 3 carbon from here and 3 carbon from here. You get 2 molecules of lactic acid. Take care of it. Sound ochindi. So this is glucose molecule breaking into lactic acid. So lactic acid is formed in the muscle cells. Next comes aerobic respiration. So aerobic respiration, we see the use of molecular oxygen for the oxidation of food. So this aerobic respiration is found in almost all the plants and animals. So complete oxidation of one molecule of glucose can be represented as C6H12O6 plus 6 molecules of oxygen, giving rise to 6 carbon dioxide, 6 water molecule and ATP molecule. So whenever there is complete oxidation of glucose, we can achieve 38 molecules of ATP if there is aerobic respiration. So on the other hand, when there was no oxygen, this glucose molecule got broken down into either ethyl alcohol plus 2CO2. The energy that is liberated in this case is 2 ATP molecule, or it can also be breaking into lactic acid, 2 molecules of lactic acid. So we also utilize this anaerobic respiration process, and the common usage is for fermentation. So this is alcoholic fermentation, this is lactic acid fermentation. So in alcoholic fermentation, the yeast is used to derive the ethyl alcohol. The carbon dioxide released helps in making the bread spongy. So here this is incomplete breakdown of the glucose molecule, whereas when complete breakdown is occurring, we find that carbon dioxide is released, water is released and energy is released. So here we get more amount of oxygen. So what will be then the differences between the aerobic respiration and anaerobic respiration? So have a look at the aerobic and anaerobic respiration. So aerobic respiration is the process of breakdown of food in presence of oxygen. So, anaerobic respiration is the process of breakdown of food in the absence of oxygen. So, this is aerobic and this is anaerobic. So, what are the end products? The end products here are carbon dioxide and water. This is carbon dioxide, this is water. Where? In aerobic respiration. In anaerobic respiration, it can be ethyl alcohol, it can be lactic acid. So, you are getting also lactic acid. So, the aerobic process will take a longer time to release energy because it will consist of more number of steps to carry out the process, whereas the anaerobic respiration path is shorter and it will be completed much faster. So, the amount of energy liberated is higher in case of the aerobic respiration, and the amount of energy liberated as compared is small in case of anaerobic respiration. So, that can be seen here. We get 38 or 36 molecules of ATP by complete breakdown of glucose in the aerobic path. In the anaerobic path, we are getting only 2 ATP molecule. So, where does aerobic respiration occur? In almost all plants as well as animal. Where does anaerobic respiration occur? Mainly occurs in species of Saccharomyces or yeast. In case of bacteria, some anaerobic bacteria like the methanogenic bacteria, and in the human muscle cell during muscle fatigue. So, when there is decrease of oxygen in the muscle, there the muscle cells also start anaerobic respiration producing lactic acid. So, these are differences between aerobic respiration and the anaerobic respiration path. So, we shall go to the details of the aerobic respiration and how it occurs inside a cell. So, this part of the respiration that we are discussing is basically known as the cellular respiration. So, it is known as the cellular respiration because we are using it inside the cell, that is, cell is utilizing this path to derive energy in the form of ATP molecules. So, coming to the steps of respiration. So, what are the different steps of respiration? So, the different steps of respiration is, we can have the steps of respiration will be, first is glycolysis. So, glycolysis is the first part. So, what is glycolysis? When we take the term glycolysis, glyco means sugar. We all know lysis means breakdown. So, glycolysis is the breakdown or the breakage of glucose molecule or sugar molecule. So, in respiration, the first part includes the breakdown of the sugar or glycolysis. So, what is glycolysis? It is the sequence of reactions which convert glucose molecule into pyruvate. So, glucose molecule is a 6-carbon sugar. What you get is pyruvate, it is 3-carbon sugar. So, you get 2 molecules. You will be getting 2 molecules of 3 carbon pyruvate. So, this path of glycolysis is common to both aerobic as well as anaerobic respiration. So, what we mean by common path is the conversion of glucose into pyruvate occurs in both aerobic as well as anaerobic respiration. So, after pyruvate is formed, the breakdown of pyruvate will depend whether oxygen is present or not present. So, this path of respiration will not require any oxygen. So, no oxygen requirement is seen. So, where does it occur? It occurs in the cytoplasm of the cell. So, here mitochondria is not involved. The entire path will take place in the cytoplasm itself. So, who worked out the steps of respiration? The steps of respiration was worked out by Embeden, Meyerhoff and Paranas. So, this glycolysis path is also known as EMP path. So, it is also known as EMP path under the name of Embeden, Meyerhoff and Paranas. So, this path here consists of 10 steps. The glycolysis path will consist of 10 steps. So, these 10 steps can be divided further into phosphorylation of sugar, then the splitting of sugar and the formation of pyruvate or the payoff phase. So, you will have phosphorylation of sugar, that is addition of phosphate, then the splitting of sugar or the breaking of sugar and the payoff phase. So, going to the details of the steps of glycolysis, first let us go to the step which is called phosphorylation of sugar. This is the first part of glycolysis, phosphorylation of sugar. So, what is our respiratory substrate? Is glucose. So, in the first phosphorylation, glucose will take up a phosphate from ATP molecule, get converted into glucose 6 phosphate. As you can see in the slide, this is glucose molecule. The 6th carbon is now getting a phosphate from the ATP molecule. The enzyme that is utilized here is hexokinase. So, kinase are the enzymes that will take out phosphate from ATP added to the glucose molecule. So, you will get glucose 6 phosphate. The second step is glucose 6 phosphate will get converted into isomer which is fructose 6 phosphate. So, this is isomerization reaction, fructose 6 phosphate. The enzyme will be phosphoglucoisomerase. So, here basically the enzyme is isomerase. So, the third step is again a phosphorylation step, which is fructose 6 phosphate getting converted into a molecule of fructose 1,6 bisphosphate. So, fructose 1,6 bisphosphate. So, in fructose 1,6 bisphosphate, you have a phosphate at the first carbon and also the sixth carbon in the fructose molecule. So, the enzyme here is, from where it will get fructose? From one molecule of ATP. So, the enzyme will be phosphofructokinase. So, you have to remember kinases are enzymes that help to add phosphates from ATP molecule. So, here we see there are 2 times phosphorylation. One, when glucose is converted into glucose 6 phosphate and fructose is converted into fructose 1,6 bisphosphate. So, what happens? So, this is the phosphorylation of sugar part. Second part will be splitting of sugar. So, in splitting of sugar, there will be breakdown of fructose 1,6 bisphosphate. Fructose 1,6 bisphosphate will break down into 2 molecules of, one is glyceraldehyde 3 phosphate, second is dihydroxyacetone phosphate, that is glyceraldehyde 3 phosphate plus dihydroxyacetone phosphate. So, fructose is 6 carbon sugar. It is splitting into 2 molecules of 3 carbon compounds, glyceraldehyde 3 phosphate and dihydroxyacetone 3 phosphate. The enzyme that is required for this process is aldolase. So, in presence of enzyme aldolase, the fructose 1,6 bisphosphate molecule gets divided into 3 carbon glyceraldehyde 3 phosphate and 3 carbon dihydroxyacetone phosphate. So, here a reversible reaction takes place. Dihydroxyacetone phosphate gets converted into another molecule of glyceraldehyde 3-phosphate. So here what is the enzyme? It will be again isomerase. It is triose phosphate isomerase. So isomerization reaction, this is a ketone sugar 3 carbon, this is aldehyde compound 3 carbon. So functional group is changing and there is isomerization reaction. So in the next steps we will go for the payoff phase. So this is the splitting of sugar part, then the payoff phase or production of pyruvate. Production of pyruvate. So in production of pyruvate, what will be the first reaction? Glyceraldehyde 3-phosphate is getting converted into 1,3-bisphosphoglycerate. So here glyceraldehyde 3-phosphate gets converted into how many molecules? Glyceraldehyde 3-phosphate is present now, 2 molecules. Gets converted into 2 molecules of 1,3-bisphosphoglycerate. So this is 2 molecules of 1,3-bisphosphoglycerate. So 2 glyceraldehyde 3-phosphate getting converted into 1,3-bisphosphoglycerate. So here we find what changes is glyceraldehyde is getting converted into glycerate. So glyceraldehyde, aldehyde is getting converted into glycerate. So what are the changes that occur? 2 NAD plus will get converted to 2 NADH plus H plus. So 2 glyceraldehyde 3-phosphate is now losing hydrogen and this hydrogen is accepted by NAD, that is nicotinamide adenine dinucleotide, and it is getting reduced. So we also observe there is addition of another phosphate group in the first position. So the addition of another phosphate group comes from the inorganic phosphate present in the medium. So here you can also see this is 1,3-bisphosphoglycerate molecule. So this molecule is 1,3-bisphosphoglycerate. So glyceraldehyde has been now converted into 2 molecules of 1,3-bisphosphoglycerate. So we shall proceed further. What happens to 1,3-bisphosphoglycerate? So 1,3-bisphosphoglycerate is now getting converted into 3-phosphoglycerate. So it is getting converted into 3-phosphoglycerate. So 3-phosphoglycerate means the glycerate, the phosphate compound is getting liberated from the first position. So this first position will be losing the phosphate. So, the next reaction will be 2 molecules of 1,3-bis-phosphoglycerate gets converted into 2 molecules of 3-phosphoglycerate. So, it is getting converted to 2 molecules of 3-phosphoglycerate. So, 2 molecules of 3-phosphoglycerate, the enzyme is phosphoglycerokinase. So, here you can see that 1,3-bisphosphoglycerate getting converted into 2 molecules of 3-phosphoglycerate. The phosphate that is liberated from here gets attached to 2 molecules of ADP to become 2 molecules of ATP. So, here 2 molecules of ADP will be taking up the phosphate from this compound, and then 2 molecules of ATP will be formed. So, this is the next reaction or the substrate level phosphorylation process. Here also again the 3-phosphoglycerate reaction, the 3-phosphoglycerate molecule will be next converted into 2-phosphoglycerate. So, in the next reaction we shall take 2 molecules of 3-phosphoglycerate getting converted into the 2 molecules of 2-phosphoglycerate. So, it will become 2 molecules of phosphoglycerate. So, here the enzyme is phosphoglycerate mutase. So, in this reaction we are not losing or gaining anything, but here only there is a shift of position of phosphate. The phosphate present at the 3rd position of this compound. So, this is the 3rd position of the compound. It is getting converted into 2-phosphoglycerate, where the phosphate is present at the 2nd position of the compound. So, 3-phosphoglycerate getting converted to 2 molecules of 2-phosphoglycerate. So, the enzyme is phosphoglycerate mutase. So, there is shift in the group. So, it is mutase enzyme that takes place. So, 2-phosphoglycerate molecule in the next step is getting converted into 2 molecules of 2-phosphoenol pyruvate. So, here you can see 2-phosphoglycerate getting converted into this molecule which is called 2-phosphoenol pyruvate. So, the next step is 2 molecules of 2-phosphoenol glycerate getting converted into 2 molecules of phosphoenol pyruvate. So, getting converted into 2 molecules of phosphoenol pyruvate. So, in phosphoenol pyruvate, so you can see this is the 2 molecules of phosphoenol pyruvate, water molecules are released, and the next step is 2 molecules of phosphoenol pyruvate get converted into 2 molecules of pyruvate. So, in the next reaction, 2 molecules of phosphoenol pyruvate in presence of enzyme pyruvate kinase will get converted into 2 molecules of pyruvate. So, this is 2 molecules of pyruvate. So, here the 2 phosphoenol pyruvate molecules are losing the phosphate each. So, here phosphate is taken out. So, this phosphate is taken up by 2 ADP molecules. So, what you will get? You will get 2 molecules of ATP here. So, here this will be 2 molecules of ATP, 2 molecules of ATP liberated. The final product of glycolysis will be 2 molecules of pyruvate. So, coming to this step up to the formation of pyruvate is common for both aerobic as well as anaerobic respiration. So, in the entire reactions of glycolysis, we can see how many ATP have been taken up, how many ATP have been liberated. So, starting from the first reaction, during the phosphorylation of sugar, we find that 2 molecules of ATP were utilized. One, when glucose is converted into glucose 6-phosphate. So, in the first reaction, we find that 1 ATP is used. So, here 1 ATP is used. Second ATP is when fructose 6-phosphate is converted into fructose 1,6-bis phosphate. So, here again we are using 1 molecule of ATP. So, how many molecules of ATP we are using is 2 molecules. 2 molecules of ATP are being used. Then how many ATP are actually produced during glycolysis? So, here we can see that there is production of 4 molecules of ATP. So, here 2 molecules of ATP when there is conversion of 1,3-bisphosphoglycerate plus 2 ADP. Here again the first position phosphate is lost, it goes to ADP, becomes a molecule of ATP. So, as we have 2 molecules of 1,3-bisphosphoglycerate, the number of ADP used will be 2 and the number of ATP released will be 2. So, here we are gaining 2 molecules of ATP. So, here we gain 2 ATP. Where else do we gain ATP molecule? Is in the last step. So, this is again 2 ATP molecule. So, total how many ATP we have gained is 2 plus 2 molecules of ATP, that is 4 ATP. So, from glycolysis the direct formation of ATP is 4, then the use of ATP is 2. So, the net gain of ATP will be 4 ATP minus 2 ATP equal to 2 ATP. So, the direct gain of ATP from glycolysis is 2 molecules of ATP in various steps. Then NADH molecules. So, here we are getting 2 NADH molecules. These NADH molecules can be used to produce extra ATP only if pyruvate enters into aerobic respiration. If there is aerobic respiration, the NADH molecules formed in this step can also liberate extra ATP. So, NADH1 molecule we shall later see can release up to 3 molecules of ATP. So, if that is the case, then if there is aerobic respiration, 2 NADH molecules will release an extra ATP of 6, and then the total gain of ATP if there is aerobic respiration can go up to 8 molecules of ATP. But how is this path important for respiration? This entire process of glycolysis will be common for both aerobic and anaerobic process. The fate of pyruvate will depend on whether O2 is present or not. If O2 is not present, then pyruvate gets converted into your ethyl alcohol or lactic acid. If O2 is present, then it will further move on. And what will happen? There will be breakdown of pyruvate. So, in breakdown of pyruvate, the fate depends on the presence or absence of oxygen. In absence of oxygen, pyruvate gets converted into ethyl alcohol or lactic acid. If oxygen is present, there will be oxidation of pyruvate. So, what happens in oxidation of pyruvate is, this is a pyruvate molecule, a 3-carbon compound pyruvate. Then this is the oxidation reaction. NAD gets converted to NADH, gains hydrogen. From pyruvate, 1 carbon dioxide is removed. So, decarboxylation also occurs. Coenzyme A gets attached. So, the remaining number of carbon here is 2 and these 2 are the remaining carbon number, and so you get acetyl coenzyme A. So, if oxygen is present, then pyruvate will enter into the next cycle which is the TCA cycle or the Krebs cycle by getting converted into acetyl coenzyme A. So, in our next class we shall further discuss of what happens to acetyl coenzyme A, how it enters into the Krebs cycle, and then how ATP is produced by terminal oxidation. So, thank you all for listening patiently to today's class.